Okay, good afternoon, everyone. Welcome to our masterclass in protein digestion, absorption models, and allergenicity assessment. My name is André Brodkorb. I'm based here in Teagasc Moor Park in Ireland, and it is a great pleasure to introduce my co-hosts. We start with my colleague Linda Giblin. Would you mind showing yourself there? My colleague Linda Giblin, also here from Teagasc Moor Park. She will be a second speaker. The third speaker will be Clare Mills from University of Surrey. The fourth speaker will be Chiara Nitride from the University of Naples. And the last but not least speaker, Corentin Lannuzel from AgroParisTech INRAE in France. So without any further delay, I will just introduce the project to you, and that's the project is, give me a second, is a Giant Leaps project. It's a European project funded by the European Commission. So Giant Leaps stands for Gap Resolution in Safety, Nutritional, Allergenicity, and Environmental Assessment to promote alternative protein utilization and the dietary shift. We have a lot of partners, 35 partners altogether, mainly in Europe and one overseas. It's a 4-year project. We are really at the end of the project now. It started in September 22 and will finish later this year. The topic of the project is really to accelerate the protein transition and make the food system more sustainable and healthier. But of course, it is pretty complex. It's a complex task. So our solution is, first of all, fill some of the knowledge gaps, in particular in alternative protein sources, drive innovation, improve sustainability, safety, and health of future diets. And also the ambition would be to achieve 50% of the total protein intake from plant and alternative sources. So the project has 10 work packages, and today our speakers are from work package 3, led by Clare Mills, Safety by Design, and work package 4, digestibility and health. And I just put in this slide. Some of you may be familiar with this. Others, if you are interested in more results, please go to the website, Giant Leaps. So these are our protein sources. That's the short list. So some of them, are low-hanging fruit in a way, fava bean, lentils, but also oat, quinoa, rapeseed. We have a microalgae here, a single-cell protein. We have crickets, an insect, and at the very end, we are also dealing with cultured beef. So we have 5 speakers, as I said, all from excellent institutions and groups. So I will start off with in vitro food digestion models, followed by Linda on absorption methods. Clare Mills will give you a good introduction of in vitro digestion and allergenicity risk assessment, followed by Chiara Nitride on some new research on anti-nutritional factors. And Corentin, he will talk about the in vivo assessment of protein digestibility. So as I said, my name is André Brodkorb. I'm based here in Teagasc Moor Park. We are a public research institute in the south of Ireland, not far from Cork. And I'm interested in food digestion in general. So why does some food behave like here on the left-hand side, highly soluble whey protein, very fast digestion, whereas other foods here, there's commonly eaten breakfast cereal, Weetabix, that's what it looks like in vitro, and that's what it looks like in vivo. And we are interested in finding out whether food structure matters and how does it influence the mechanism and the kinetics of food digestion. So my general interest is in food digestion, but one big topic is protein digestibility. So protein digestibility is basically a number, okay? And here are some of these different methods, how to determine them. Some are very old, some are inaccurate, some are better than others. So that was nicely summarized by a recent review. But to determine the protein quality, it generally requires in vivo study, right, for regulatory and labeling purpose. And generally that is done either in rats, pigs, or humans. So when you look at rat models and pig models, you would question the relevance, especially with the rat rodent models. The pig models, they are certainly better, but there again, you have the question, do you have to slaughter dead or alive, or like here in the middle, you have cannulated pigs, which is a very good model. But again, you have ethical questions. Is it relevant? They're expensive. And then blood and guts. So as you know, animal trials are not as popular as they used to be. So some of the companies that we are dealing with, they don't want animal trials anymore. So we are lucky that we have an excellent group in AgroParisTech in this project. So Corentin will cover the human intervention trials, and that's really the gold standard. When you look at protein quality or digestibility, as I said, there are several different methods out there. An older method would be Teagasc. A more recent and more accurate and recommended method would be the Digestion method. So it's digestible, indispensable amino acid score. And here we are looking at the digestibility at the end of the ileum. Okay, and in general, that's done either when you slaughter pig or cannulation, or in terms of for humans, you use a tube, and Corentin will cover that. So human models, the human model is certainly the gold standard. There's no doubt about it. The animal studies, give me a second now. Here we go. The animal studies, they are good, but they are certainly a second choice. They're also expensive. Relevance could be questions, and time, slow basically. So there is a drive to replace, reduce, or refine it, especially to replace it. So there is a place for in vitro lab-based models, right? So in vitro lab-based model in general, they are relatively simple. They can be quite crude because they don't have this feedback loop that you would have in vivo. Okay, so when you look at the different digestion models, first of all, you can there's a case for not doing any digestion. So if you eat 1 gram of protein, that's what it is. Do you write that on a packet? It's 1 gram of protein. If you drill a bit deeper, you have different methods, and they all vary in ease of operation, how crude they are, or how refined they are. And some of these are very accurate, but also very complex and very expensive. So I just want to show you a number of these models. So there are some very sophisticated dynamic digestion models. So the most well-known is probably the TIM model. That's the older TIM1 model developed in TNO in the Netherlands. So here you see the stomach and the small intestine here. They have an absorption cell, which is a very good simulation. There are other models out there, about 10 to 12 models. There's another one here in France, or the recently developed model from China. If you're interested in these dynamic models, a number of years ago, 2022, I was hosting a webinar where the 11 available dynamic models, they all had a time slot of 20 minutes. So if you're interested in these, go to our YouTube channel. So these dynamic models, they're good models. It's a good representation of the in vitro digestion. Disadvantage would be access. So you can only run 1 or 2 samples per day, because that's what it takes. They are in general commercial, or at least semi-commercial. So there's a commercial interest in selling the instrument. They are not standardized. Okay, they're not standardized, so the conditions, they are very often proprietary, and they are expensive to purchase, and they're very expensive to run. Okay, so there is a case for simpler models, and there are a lot of simple models, and I want to concentrate on one particular type of model, and they're called static in vitro digestion models, and they are called so-called one-pot model, where the three digestion phases, the oral, gastric, and intestinal phase, are simulated in one pot. And the model that I want to talk about is the InfoGest model, and that came out of a European EU-funded cost action. So InfoGest stands for Sharing Information on Food Digestion, and myself and my colleague Isidra Recio from Madrid were leading one of the work packages, working group, to find international consensus. And that's what it looks like, really a one-pot model. So you have basically one tube. You simulate the oral phase with all its ingredients. Then you change the condition in the same tube to simulate the gastric phase. You add the pepsin, if necessary, the lipase. You change the pH to pH 3 over 2 hours. And then after 2 hours, you change the conditions again in the same tube to simulate the intestinal phase. So you add your pancreatin, you change the pH, you add bile, and so forth. And at the end of it, you have to do your sample preparation for your downstream analysis. So that was published initially in 2014 in Food and Function, and then in 2019 in Nature Protocol, where we really set out a protocol, a step-by-step protocol with all the individual steps outlined, including troubleshooting. So that is an international consensus, and it's based on available physiological data. Some of the data is just not available, so it's a best guess. And now for the last few years, that's really the academic and industry standard for static in vitro digestion methods, and we define enzyme assays in the protocol. We give some recommendations of the downstream treatment. Is really case by case, depends on what you want to look at. If you want to look at food structure or bioaccessibility, we give some recommendation. We also provide some tools, so spreadsheets, how to perform the in vitro digestion. There's a YouTube channel at Food Digestion, one word. We had some training schools, and it has already been used and published thousands of times already. So following on, we further developed the method based on the InfoGest method to look at the protein digestibility. So that is currently happening for a IDF ISO method for the assessment of protein digestibility. So that was led by Lotte, Isidra, and myself. And only a few weeks ago, Lotte Egger and Reto Portman from AgroParisTech, they were leading this particular study and was published in Nature Protocol. It's called InfoGest Quant. That's basically based on the InfoGest method, and then the downstream analysis to simulate an in vitro DIAS method. So these static models, they're pretty good. They're standardized. They're simple but not easy. They're good for screening, and they're good to estimate endpoints, so the end of gastric phase or the end of the intestinal phase. Where they are not so good is for kinetics. Okay, so one-pot models are not suitable to estimate the kinetics of food digestion. And that's where the dynamic models come in. But as I mentioned earlier, dynamic models, the availability or accessibility is not that great. So a number of years ago, we came up with a standardized semi-dynamic digestion method. Semi-dynamic because the gastric phase is dynamic and the intestinal phase is static. And we used commercially available equipment, titrator, syringe pumps, the vessels, and here we basically simulate the change in the gastric phase. Here, in this case, is the gastric volume, but the same for pH. You start with a high pH, and bit by bit, the pH changes, and more enzyme is added, and the gastric volume also changes because we simulate the gastric emptying points. So as I said, we use commercially available equipment, including the stirrer that can be 3D printed. So that way, whether we perform the digestion here in Ireland or in the UK or in Australia, we can actually compare the results. And you see something like this. This is an example from Annabelle Mollet-Cabero's work, where we can see the digestion of, in this case, it is 100% whey protein as a function of time. So we can actually see the digestion over time, look at the kinetics, and you can see fine differences. And these differences would be very hard to see in simpler static digestion models. So I think that's my last slide. That's where we are at the moment. So we have a static digestion model for adults. That's the international consensus. There's also a model for older adults, elderly, and a well-established infant method. In terms of semi-dynamic, we have established international consensus on the adult, and there's currently in progress an infant model and a model for older adults. So I would like to thank you, and we move on to our next speaker, my colleague Linda Giblin here from Teagasc Moorpark, and she will talk about in vitro absorption models. Over to you, Linda.
Hi, everybody. So I'll just share my screen.
Yeah, that looks good.
Pointer. That looks good. Okay. So, as André said, my name is Linda Giblin, and I'm from Teagasc as well, and I'm going to talk to you about in vitro methods to assess specifically protein absorption. So, the first thing is, what is absorption? So this is when nutrients or food compounds are transported from the gut lumen across the gut barrier. So for digested proteins, that means small peptides, di, tri, and tetrapeptides, and free amino acids. So they must cross from the gut lumen, they must cross the gut barrier into the bloodstream. So once nutrients cross the gut barrier, they're bioavailable. And true bioavailability is, of course, when the nutrient reaches the target cell, organs, or tissues. So where is proteins and amino acids absorbed? So they are absorbed in the small intestine, and the small intestine is made up of the duodenum, the jejunum, and the ileum. The duodenum is closest to the stomach, and the ileum closest to the colon. So the protein is digested from your food in the small intestine into these peptides and free amino acids. And the small intestine then is 6 meters in length. So it's quite long, but it has a much more increased surface area because of these worm-like projections or villi. So its surface area is 250 meters squared. So it's huge absorptive capacity. So this is what the villi look like. So here you will have the gut lumen, and the nutrients have to cross a single layer of cells so they can be taken up by the lymph system or blood system. And this is just a histology slide of pigs here in Teagasc, and you can see that this is the duodenum. This is what they look like, these villi in the duodenum. So let's talk about this gut barrier. So this is the gut barrier here. It's a single layer of cells, and it's made up of 4 different types of cell types. And you can see that there's a green layer over it. It's green here in this diagram. It's not actually green, but this is the mucus layer, and this protects those cells from the gut lumen contents. So the 4 different types of cells are the enterocytes, which are the main primary cells in this gut barrier. They are the absorptive cells. They're most abundant, and they occupy at least 80% of the gut barrier. Then there are goblet cells, which are about 4 to 12% of the population, and they produce that mucus layer which protects the barrier. Then there are enteroendocrine cells, which is about 2% of the population, and they produce satiety hormones. How full do you feel in response to the food that you're eating? And then there are pancreas cells, which are about 2% of the population, and really they produce digestive enzymes, antimicrobial peptides, cytokines, and they're down here in the crypt because they maintain the health of the villi. So, how is nutrients absorbed across this gut barrier? Well, the first thing I would say is that the individual cells in the gut barrier hold hands with their neighbors by tight junctions. So there are two mechanisms in which nutrients can pass from the gut lumen to across this barrier. So paracellular is where they go through these tight junctions. Transcellular is where they go through the cells, and that can be passive or it can be active, where they're taken up by a transporter or a carrier and brought across to the other side. So if we specifically look at peptides and amino acids from the digesta, they will primarily use an active transcellular transport. Like, for example, PEPT1 is the peptide transporter, and there are also various amino acid transporters on both the apical side, which is the gut lumen side, and on the basolateral side. So what are the methods to create gut barriers so we can do absorption experiments? Well, they're primarily cell-based, obviously, so we can look at intestinal cell lines, or we can grow or culture organoids in a two-dimensional or three-dimensional shape, or now you'll see these guts on a chip. But there are also artificial ways we can do it. We can do it by using artificial membranes, or we can use ex vivo, which is biopsy samples from animal tissue, where we can set up Ussing chambers, or indeed now we can do in silico, where we can predict, knowing the permeability characteristics of the nutrient and knowing how the barrier operates, we can predict what the permeability would be like. But I'm just going to concentrate on cell-based in this presentation. So in the cell-based methods, you have either intestinal cell lines, and they can be grown as monocultures into a barrier or as co-cultures, and there's a wide range of those cell lines, but the most popular are CACA2 and then HT29MTX. There are also organoids, as I said, and there are also the gut on the chip, which introduces the flow of nutrients. So let's get back to the first one, intestinal cell lines. As I said, the most popular are CACA2. These are derived from human colorectal carcinoma from a 72-year-old Caucasian male back in 1977. And what they found was that when they grow these cells in these transwell plates for 21 days, they'll actually form a polarized barrier, which has tight junctions. It has secretory vesicles on the apical side. It has microvilli. It has a brush border. So are these CACA2 barriers suitable for protein absorption? So this table lists all of the peptide transporters that we know, the apical amino acid transporters and the basolateral amino acid transporters. And here is the expression levels in the small intestine, both mRNA transcripts and protein by Western blots. And you can see that there are different levels of expression, but they're all present. And here are CACA2 cells, and you can see that they have all the peptide transporters. On the apical side, they have all but one of the amino acid transporters, and on the basolateral side, they've all but two. So they do have practically the full complement of peptide and amino acid transporters, although expression levels may differ within vivo. So what is the method to create a polarized stable CACA2 monolayer? The first thing that you must do is obviously take it out of liquid nitrogen storage and resuscitate it. And we recommend that you do 3 passages of this in a flask first. We usually work between passage numbers of 20 to 30, and the media is usually DMAM plus 10% FBS, fetal bovine serum, plus antibiotics. And then after 3 passages, you're ready to seed into transwell plates. We recommend a low density seeding so that you ensure that you will produce a monolayer. And so we do about 6 by 10 to the 4 cells per well in a 12 well plate. Then we allow these to culture for 21 days, but you have to replace the spent media every 2 to 3 days. And the volumes you're talking about is about 0.5 mils in the apical and 1.5 mils in the basolateral. And the last step then is, as they are growing for the 21 days, you have to monitor that a barrier is being formed. And you can do that by electrical impedance, which is transepithelial electrical resistance. And you measure that every 7 days, and you want that to increase over time. But you never do this measurement on a day in which you're changing the media. Now, when day 21 comes, you then have to make sure that the barrier is formed. So for this particular Caco2 clone HTB37, you'd expect your TER values at day 21 to be above 800 ohms by centimetre squared. You want to make sure that the barrier is stable, that there's no further increase in TER values. Often at day 21, people then also run a barrier permeability test. They make sure that the paracellular transport is working and the transcellular transport is working. And they do that by using fluorescent compounds like lucifer yellow or with pharmaceutical compounds. You can also check that the barrier is mature, and you can do that by the expression of brush border enzymes, for example, alkaline phosphatase. So what does the absorption experiment look like then on day 21? Well, here you have your food, and as André talked about, is you can then do an in vitro digestion with the InfoGest method, so that your protein in the food is now digested into peptides and free amino acids. You add that food to the apical side of your polarized monolayer that has been grown for 21 days, and you do the incubation for about 2 to 4 hours in a simple buffer like HBSS with an energy source or simple media. And then what you want to do is you want to see that the peptides and amino acids are crossing from the apical chamber into the basolateral chamber. And what a word of warning is that you need to apply sufficient food digestive material here so that you can detect it in the basolateral, and you can detect the individual amino acids and the peptides in the basolateral. Now, that will really depend on the type of instrument you're using for detection and its detection limits. So before you start, however, there are biocompatibility issues between food digester and cell monolayers. And the main challenge is that the food digester contains digestive enzymes, for example, trypsin. And trypsin will lift your cell monolayers. Bile salts are toxic, and there could also be osmolality issues. So the solutions are to remove or inactivate the digestive enzymes and to dilute the salts before you put the food digester down on your monolayers. The warnings, however, are that if you use enzyme inhibitors, they can often be toxic to your cells. They can inhibit brush border enzymes, and the alternative would be to heat the food digestive sample, but then you can include structural changes in your food sample. You can remove the enzymes by using, for example, molecular weight cutoffs, but then you could also remove food components. And you can dilute, but then you may be back to being unable to detect your nutrient in the basolateral chamber. But we do recommend that you do a biocompatible check pre and post a detoxification. And what that means is you take your digester sample at different concentrations and you incubate overnight cells with this, and you run just simply a cell viability assay like an MTS assay. So we have recently published a paper back in 2024 on a series of recommendations which give you a step-by-step guide depending on your research question, type of digester and food component. So you'll be able to make decisions on how you're going to detoxify your food digester before you put it down on your cell monolayers. And it is something like this. This is a graphical of a yes, no, yes, no. So what are the advantages with Caco2 cells? Well, I suppose the first advantage is it does have good permeability correlation with human gut. It has an R-squared of about 0.84 with 21 drug components. It is tried and tested, so there's lots of data in the literature. It is commercially available. It's quite a robust cell line. It's easy to culture. If you do 12 well plates, you can do 3 plates per day per person. So it's not high throughput, but you can get through a number of samples. It is suitable for food digester, provided you clean it up. And there is an option to add additional cells directly to the monolayer, like HD29, or to the basolateral side, like, for example, if you want to add M cells. And it does express the transporters, particularly for peptides and amino acids. It seems to have almost the full complement of those transporters. The limitations are that it does have narrow, tight junctions compared to human gut cells. So you'll see that CACA2 cells are about 0.4 nanometers between each individual cell, where it's about 1 to 1.3 nanometers for human cells. It also has the electrical resistance over it is greater than 800 ohms by centimeter squared, whereas with humans it's much less, it's 50 to 100 ohms by centimeter squared. Absorption is influenced by solubility, by hydrophobicity, polar surface of the compounds. They are cancerous cells, so they do not use butyrate as an energy source, and butyrate would be the main energy source of intestinal cells. They lack the metabolism enzyme cytochrome P450, and then they don't have a mucus layer, and we know that mucus is present in the intestine, and there's substantial interlab variability due to different protocols and different CACA2 clones. So the last two there, I'll show you what we have done to remedy that. So many labs now they add goblet cells without disturbing the CACA2 monolayers. So this goblet cell, this cell line HD29-MTX or HD29 was derived from a human colon adenocarcinoma from a 44-year-old female. And they found that if they treat it with methotrexate, the subclones mature into actual goblet cells producing mucus. So what people do now is they often seed the CACA2 cells with these HD29-MTX at a ratio of 90 to 10. And then they allow the 21 days to form the polarized barrier, and it will have a mucus layer. To overcome the variability issue within Giant Leaps, we set up an international ring trial where we agreed protocols on the CACA2 HD29-MTX culturing, on seeding the transwells, on measuring the TER, on measuring the paracellular and transcellular permeability. So this was across 8 European countries, across 17 laboratories. And the outcomes of that are that we want to reduce the variability via these consensus protocols, much like what André was talking with the Infogest standardized protocols. We also want to be able to identify a stop no-go where you have to have a minimum TER at day 8 or that barrier will never form by day 21. So it gives people a chance to stop their experiments. We also can now define a healthy versus an inflamed barrier, and we can try to understand and unravel the relationship between electrical impedance and also how that TER value relates to lucifer yellow permeability. So just then to mention the other types that are out there, I'm not going to mention methods for these. They are the organoids and the gut on a chip. The organoids, the 2D organoids, our intestinal epithelial barrier is formed starting from stem cells, and they will have the full complement of cell types. They'll have a mucus layer, enterocytes, goblet cells, pantheate cells, stem cells, enteroendocrine cells, and tough cells. And it's a complex protocol, however, to start with stem cells and differentiate it into intestinal epithelial cells. But you do have multiple cell types there. You have a cytochrome P450, and you do, because you're growing them as a monolayer in a transwell, you do have an accessible apical side. If you go to 3D organoids, that's talked about, again, we talk about taking stem cells, usually from crypts actually, and they will self-organize into intricate tissue-like structures which are 3-dimensional, and therefore they mimic the architecture of the intestine. However, there is an accessibility issue to the apical side. So now they've done an apical in-enteroid and an apical out version. What additional characteristics will got on a chip? These are now commercially available, and what they actually will be is they will have intestinal epithelial cells, but they'll also have a microfluidic device that mimics fluid flow and therefore nutrient movement, and that simulates peristalsis. So that's that additional characteristic to add to the system. So just to recap, this is your absorption experiment. You start with food, you digest the food. In the background, you grow your cell lines, or you differentiate your cell lines into a polarized monolayer. Then you add your digester to one side of the chamber and you measure what crosses. So that is nutrient absorption. So thank you very much.
Excellent. Thank you. Thank you, Linda. So our next speaker will be Clare Mills. I just forgot to say, if you have any questions, type them in the question and answer box. Would be actually good if you have any question, if you type them now, because we can see them then during the talk. Okay, very good. Over to you, Clare.
Yeah, and just to say, Chiara is also contributing to what I'm talking about today. So let's get going. All okay, André?
Yeah, perfect.
Okay, so in vitro digestion and allergenicity risk assessment, which is in work package 3 of the Giant Leaps project, and I'm going to be talking to you about this today together with Chiara Nitride, who also contributes to this work in Giant Leaps. So hopefully the slides will move on. First of all, my declaration of interest, just to say that I do work for the UK Food Standards Agency, and I'm on the Advisory Committee on Novel Foods and Processes, and I do work for the European Food Safety Authority. And I leave this disclaimer in because some of the work that we talk about has also been done outside the framework of Giant Leaps, but under a EFSA procurement. So they require this disclaimer to be added. So just going back to the whole concept and why digestion is important in allergenicity, is that hydrolysis is acknowledged as a way of removing the allergenicity of foods. So, and I don't quite know how my mouse was moving without me doing anything, but anyway, there we go. So small peptides lack the multiple sites that are required to elicit an allergic reaction. In order to cause an allergic reaction, you make a molecule called IgE, which is normally produced to help protect you from parasitic infections. And in some people, for some reasons, they begin to make this towards their food. But those antibodies have to be directed towards multiple epitopes. I'm not sure where are there 2 pointers on the screen? Because there's one that seems to be moving around randomly. I'm not sure where that's coming from. I'm not used to using Zoom, I'm afraid. I don't know whether you can see mine, but there are 2 laser pointers on my screen. That's not mine. I don't know who's moving that. Anyway, we will keep going. So if you start breaking your protein down into small peptides, you lose the IgE epitopes. I have to say, I'm finding this very distracting. Completely, I take that away. I'm not going to bother. If you have enzymatic hydrolysis, it's used actually to prepare hypoallergenic ingredients. And what you can see here in the bottom left of my slide is a study done by Katrina Bowe in actual animal models, looking at the capacity of hydrolyzed beta-lactoglobulin from cow's milk whey and a hydrolyzed whey protein to cause the development of antibodies in an animal model compared to the intact protein. And basically, when you hydrolyze it, the capacity to both cause an allergy and then elicit an allergy is very much reduced. That work is also backed up with a study that was done by Nestlé with Sophie Newton, where they showed that if they hydrolyze the beta-lactoglobulin, they have almost undetectable protein, and it abolishes antibody binding. So that's the hypothesis that we work to, that if you hydrolyze your protein, you're going to reduce its allergenicity. And that underpinned the original work done back in 1996 by Jim Astwood and the guys in Monsanto, which actually took us all on this journey and the stability of food allergens to digestion in vitro. And in fact, the paper of Jim Astwood's published in Nature Biotech hypothesized that food allergens must present sufficient gastric stability to reach the intestinal mucosa, where absorption and sensitization, so the development of allergic disease, can occur. And this gave birth to something called the pepsin resistance test, which is now really considered a physicochemical marker of allergenic potential. And the test was developed using simulated gastric fluid derived from US pharmacopoeia and recipes used essentially for drug dissolution testing. And the original paper purports to show that resistance was greater for allergens than putative non-allergens. And back in 1996, we didn't know very much about food allergies, and this has basically become enshrined in legislation as part of the safety assessment of GMOs. And so we have had this integrative weight of evidence approach for looking at predicting new allergens, de novo sensitization, and prediction of cross-reactive allergies, which has included this pepsin resistance test and in vitro digestibility tests for quite some time now. So we know a lot about it. However, what has come clear over the time is that the relationship between resistance to digestion and allergenicity is not clear-cut. So this is work that I and my team did in Manchester back in 2013, looking at different purified proteins digested using the pepsin resistance test or variants of it. And really what we found was that resistance to digestion was not predictive of allergenicity alone, but it did give you a very good diagnostic readout of the susceptibility of different types of protein structure, different scaffolds. So for example, the 2S-albumins, the lipid transfer proteins, were highly resistant to digestion, whereas the BETV1 homologs, which are considered to be less problematic allergens, were way much more digested than the 2S-albumins and LTPs. So what we now have is pepsin resistance test that is really now much more considered to be a marker of biochemical stability of a protein, but gastroduodenal digestion is still valuable because it provides understanding of the context in which a protein is presented to the immune system in a physiologically relevant context. So now we have a new approach, and this is from EFSA, published I think late last year, which is now moving towards a stepwise, I love it, they slightly amend it, stepwise weight of evidence approach. But what you can see here is that our in vitro gastrointestinal digestion has very much moved into this protein stability, looking at pH, heat, and gastrointestinal digestion. And what is important is that it is now expanded not just to look at allergens, but also look at toxins and anti-nutritional factors. So how do we do that in vitro digestion? And the processes for allergenicity have really come from that pepsin resistance test, which was grossly unphysiological, to see how can we develop conditions that are more relevant to the physiological situation, and that also reflect conditions found in vivo in perhaps what we would consider susceptible groups, so infants, where we know the gut is immature, may not produce as much pepsin, may not reduce the pH down as low as you find in adults. Looking at fasted versus fed states, And also just think 25–30% of the European population will take antacids at some point, and the high, very high pH that you get in the stomach as a consequence of their use, which almost stops pepsin digestion completely. And then you have intestinal conditions again, which can reflect those perhaps found in vivo in adults and infants, where there are fewer lower levels of enzymes produced. So in Giant Leaps, one of the things we've done in Work Package 3 is really map out all of these conditions, and it's been applied in a 96 well plate format with physiologically relevant biosurfactants, because the original pepsin resistance test and the intestinal digestion test there don't take account of surfactants. So the other thing that is important, and André touched on in his great introduction to this whole masterclass, is about sampling. And for allergenicity risk assessment, we want to know not the endpoint, but actually how is the protein digested and how will it get the exposure of the immune system. So the important part of the immune system in the gut wall that's involved in sampling are the Peyer's patches. These are found in the small intestine. Most of them are found towards the terminal ileum, but they are scattered throughout the small intestine, and the first Peyer's patches are present in the duodenum. So understanding early gastric emptying events is really important. The time courses, and this is just a wonderful study from 2019 looking at the impact of calories and also the difference between particulates and soluble material and how that influences gastric emptying. And what has been arrived at, but again, like André said, with the InfoGest protocol, you come up with a calculation based on what you're hoping is going to be right. Or as an approximation, is generally we're taking a gastric digestion that at 10 minutes is the important one, because it's when you get the first gastric emptying event into the intestinal phase. So that's one of the challenges for the allergenicity risk assessment, is that we need to sample and we need multiple time points to provide data for kinetic analysis. So I'll now pass over to Chiara, who's going to pick up the baton, and I will change the slide as you tell me.
Thank you, Claire. So now we move on to showing you some applicative examples of how we can actually analyze and collect readouts from these samples that we have collecting at different time points of gastrointestinal digestion to inform about the fate of these allergenic proteins. So the first step can be electrophoresis analysis that can be coupled also with immunoblotting. So in this electrophoresis analysis, what we can do is to collect information about proteins and polypeptides above a certain molecular weight that would be around 10 kilodalton. And it is very important, and there are some considerations to make, and this is related with the type of enzymes that we actually select for simulating the gastric and the duodenal phase. And this actually will become even more clear later on, but we tend to use purified enzymes. And this is because this is crucial, as we want to follow the breakdown of the proteins electrophoretically, visually, basically. The use of this purified enzyme would actually help maximizing the clarity of the information that we are following. And another advantage of electrophoresis is that this technique can be semi-quantitative through densitometry analysis. So it means that we can actually follow persisting peptides, and we can actually calculate the half-life of specific bonds and quantify the fate throughout the digestion, the time of digestion. Next slide, please.
Ooh,
So
it's a
to
time
support to support electrophoresis, which can tell us, together with immunoblotting, whether a band is a given protein and how actually it's breaking down, what mass spectrometry can provide us is basically the name and the surname of this protein, can provide us with the sequences of the polypeptides. mass spectrometry can work out to a lower range of molecular weight, meaning that we can target much shorter peptide, as short as 4 or 5 amino acid lengths, which may not be that relevant for allergenicity risk assessment, yet is the shorter length of a peptide that can be inferred to a protein with a high confidence. As when we go down to a lower molecular weight with 2 and 3 peptides, it is more difficult to uniquely infer and associate that sequence to a protein. And then we can monitor SI can be 3000 m/z, which means lengths that can go up to 30, 35 amino acids. So quite large polypeptides. Next slide, please. So in order to increase the confidence of peptide identification, and this is true also for proteins identification through mass spectrometry, we need very strict identification criteria. We need a very high repeatability and reproducibility, because if you think about it, we are talking about a very complex system where we simulate the digestion of proteins, and we also then actually implement a downstream process to prepare the samples for mass spectrometry analysis. So repeatability and reproducibility are really, really important to make very strong the outcome of the analysis. With mass spectrometry for identification, we do rely on protein databases. This is also quite of a critical point, a critical aspect, as when we think about novel foods, alternative foods, the presence of well-annotated and curated databases is not a given. Sometimes we have to work with homology, and this can actually in some ways influence the type of readouts that we have. Nevertheless, there are bioinformatic tools which are really interesting and really help out with what we call de novo sequencing, meaning that we can, from our mass spec data, sequence peptides, and then with these peptide sequences, we can perform what is called identification biomology and still have an idea of the persistent peptides that can be identified in our food ingredient and can be associated to the ingredient itself. Once we have collected all this information, then we can move into trying to understand whether in these long peptides there is an epitope that can be persistent epitope that align with the protein sequence. And when we look at the molecular level, our proteins, and we manage to identify portion of the proteins which is more resistant to digestive enzymes, in that case, we can start understanding more in depth, for example, the influence, the sequence of the protein up to the post-translational modification that can naturally occur, how this can then influence the way the protein is broken down, and also the way actually is folded with disulfide bonds. And in this context, we can even start looking into induced post-translational modification that can be related with processing. Next slide, please. So just a few examples. What you see here, these are electrophoresis that have been performed on purified proteins. And you can see these are time points throughout the digestion, which was performed at different pHs. So also challenging actually the ability of the enzyme to degrade. And you can clearly see that it's easy to follow the breakdown of the protein throughout the time points. So we can actually build up a real kinetics in a simplistic model. Next slide, please. And thanks to this readout and thanks to densitometry that we can collect information, and we can actually collect objective information that would inform about the kinetics behind the breakdown of the proteins. We can clearly see which are the transient digestion product and which are the most persistent product that would lead to a longer exposure in the gut. Next slide, please. So we have been talking about proteins, we have been talking about purified proteins, but it is applicable also to more complex ingredients and even foods. And this is an example where we have been following the fate of allergenic ingredients in the context of a baked muffin. This was meant for oral immunotherapy to deliver agamic allergenic ingredients. And what you can clearly see here is that we can follow and monitor several different types of ingredients, allergenic ingredients, from the egg, peanut, and even milk. And we can see that in this context, even at a further stage of the digestion, we can clearly see that there is still resistance immunoreactivity. So there is still polypeptides that are more resistant. And this is actually more evident when there is a lack of the pepsin digestion. So when actually at the very early stage of gastric emptying. Next slide, please. Again, complex food matrix. Here is an application of mass spectrometry to the study of the digestion of a pizza, which is just a baked product, is a model of a baked product, where we follow the digestion of gluten proteins as well as soybean proteins. So one of the first outcomes from mass spectrometry would be the length of the peptide and the abundance, the number of peptides with that given length at that given time point of digestion. And what we can see is that during the digestion, gluten proteins would lead to much longer peptides as compared, for example, to soybean protein. And this is intrinsic to the primary structure of this protein, as gluten, of course, is much more rich in proline and amino acid that would reduce the ability of this peptide to be digested even in duodenal phase. Next slide, please. So going back to the drawbacks of pancreatine. So we tend to use pancreatine as a complex pool of enzymes for duodenal digestion. This is what is used in InfoGest. However, as I said at the beginning, pancreatine is quite of a problem when we want to use electrophoresis as one of the techniques to collect readouts. And this is because it's clearly seen on the right-hand side of the slide, where you really see in the pancreatic phase, in the intestinal phase, we can really see that the food proteins are masked by the complexity of the pancreatic enzymes as compared to, on the left-hand side, a digestion that has been performed with the purified enzymes. So how to overcome this? One of the ways to use purified enzymes, or what can be done is to use immunoblotting, where we can use actually antibodies to mark the proteins and follow the bands related to the given allergenic protein. Nevertheless, also in this case, we have a limitation. From one side, antibodies, they have to be very well characterized to have a confident readouts. And on the other side, is also true that they may fail in recognizing very large fragments, and so we may fail at monitoring properly these digestive enzymes.
So just to finish up, we've given you a bit of a big overview, and you can see how in vitro digestion has evolved over the years since 1996 in Jim Aswood's paper. And I think we have much better tools and approaches now that can help us with assessing potential allergenicity. But there are still aren't there, always gaps and needs. I mean, one of the things really important is for reporting, that people do report in their studies protease to protein ratios, providing enzyme activity units, where you actually do your studies, that you use different pH conditions and those enzyme ratios that might represent physiological conditions found in either infants, adults, or those with an impaired digestive capacity, and something that, in addition to antacids, Many older people these days are put onto statins, and they have a profound effect on steroid metabolism, including bile salts. And you also have people with issues over bile production, which may also affect digestibility quite considerably. One of the things, in addition to the 96 well plate format, we've been trying to refine the conditions for allergenicity risk assessment, and so that we can actually connect a bit more to the approaches taken in InfoGest for looking at the nutritional quality of proteins. And we're definitely coming up with harmonized and perhaps more standardized methodology. But what we're still needing going forward beyond Giant Leaps, I think, is a need for interlaboratory assessment. It provides more credibility if you show the transferability of test approaches, and using appropriate methodology for monitoring the outcomes of digestion and readouts. And we've done a lot of work to actually move towards readouts that can actually provide better interpretation of test results. And lastly, and this is something that I have a bit of a bugbear about, but maybe André also would like to see more standardization in InfoGest, is this need to include comparator and standardized proteins, and very, very few laboratories include those. And I think that is something that is going to be really important for the future use of this methodology more widely. So just to say thank you to everybody who's contributed from Work Package 3, but particularly to Giovanni Daria, who works with Chiara, and two PhD students, Charles Kunjin and Laurie Stevenson, who work with me, and their PhD projects are associated with the Giant Leaps project. And thank you for listening.
Thank you, Claire. Wonderful. So, Chiara, you're the next speaker.
Yes.
Excellent.
So I'm going to share my screen. Can you see the screen?
Yes, perfect.
Okay, perfect. So good afternoon again. I'm Chiara Nitride from the University of Naples, and I'm going to take you through exploring anti-nutritional factors in the context of the digestibility and the safety of alternative proteinaceous ingredients. So when we think about anti-nutritional factors, we are actually bridging the gap in between nutrition and safety. And the identification, characterization, and quantification is a part of the pre-market evaluation of alternative proteinaceous ingredients, whether it is part of the nutritional assessment, because they are considered nutritional limitation, or if they are considered naturally occurring toxins. Their assessment and the evaluation is actually has the final goal of providing ingredients with high quality that are nutrition and safe. So what are anti-nutritional factors? These are molecules that are able to actually interfere with natural processes of digestion, absorption of nutrients. So basically are molecules that would interfere with metabolism of nutrients. And if we think about plants and traditional ingredients like pulses and cereals, these anti-nutritional factors are actually present in these ingredients, naturally present in these ingredients. And from a plant perspective, they are chemical molecules, are chemical complexes which are produced because plants do not have legs, so they cannot run away. Therefore, they have developed a mechanism that would protect them from predators, from pests, but also from environmental stresses like can be extreme heat. So in this context, and in the context of climate change and global warming, the assessment of anti-nutritional factor becomes even more important in traditional food as well as in more alternative and novel food products. So when it comes to the classification, we have categorized anti-nutritional factors in non-proteinaceous anti-nutritional factor, primarily small molecules, and proteinaceous anti-nutritional factors. Within the non-proteinaceous anti-nutritional factors, actually, we have a lot of molecules, some that are also associated with taste and of flavors like bitterness, astringency in the mouth. And just to name a few, we have the phytic acid, which is known to bind the minerals and impair their absorption. But in the context of protein digestions, also polyphenols and dietary fiber can be considered anti-nutritional factors, given their ability of binding to proteins, and this can be both food proteins as well as the gastrodigestive enzymes, impairing their ability actually to digest macromolecules. When we look instead at proteinaceous anti-nutritional factors, one of those that we may be more familiar with are lectins. Lectins are proteins that are capable of binding sugar moieties, and these can bind to the enterocyte in the intestine, altering the natural permeability, and they are considered enterotoxins. And then we have enzyme inhibitors, both protease as well as amylase inhibitors, which are again proteins that have the ability of blocking gastrointestinal digestive enzymes from breaking down macromolecules, which we can only absorb after digestion, to small amino acids. So when we needed to approach the assessment of anti-nutritional factors within the Giant Leaps project, and we actually screened the ingredients that at very beginning André showed you, which can go from plant proteins, so plant ingredients, all the way to single-cell proteins or insects, we decided to use a stepwise approach. We started from an in silico bioinformatic analysis, where we started gathering information from literature, which may be available for more traditional alternative ingredients, but then when you move into more new ingredients, literature review can be much more limited. And then we took a step further and we approached through phylogenetic analysis. We tried to see all these ingredients, whether they could be related with more described and more characterized food ingredients. And we started digging and analyzing whether available genomic data, proteomic data, and metabolomic data. There are bioinformatic tools that can help you out when you want, for example, screen the potential presence of lectins based on genomic data that are available on organisms. So we move a step further and we kind of, after this actually review that we have done, which was an exercise that involved multiple work packages, including work package 3, we moved a step further and we started working in the lab to try to collect information about our ingredients. And this was done through proteomic analysis and metabolomic analysis, so through high-resolution mass spectrometry. So what is interesting and why this is needed? This is needed because genomic data are static data. So they tell us about the potential that something can happen within an ingredient, within an organism. But actually, proteomic and metabolomic are dynamic. They work, and protein expression, metabolites are expression of environmental events that can occur around our ingredients, around our plant, around our microorganism. So collecting at a molecular level, protein level evidence of expression or the presence of the metabolites can actually tell us how that food is, that ingredient, that organism is reacting to the environment. Now, this exercise can be challenging, and can be challenging because we need very well curated protein databases for proteomic analysis, as well as we really need well annotated spectral library for metabolomic analysis. And this is not a given, and is something we may need to work towards, particularly for more innovative ingredients. So this step is rather crucial and important because it would tell us how to move forward, meaning what are we going to do next? What are we going to analyze in the lab? What are we going to look and measure the activity for? And this is done through a combination of in vitro assessment that would quantify the molecules and also tell us the activity of the molecule. And then a step further would be assess the activity, the effect of that molecule under in vitro physiological conditions. So basically, an inhibitor, how much would inhibit the gastrointestinal digestion in vitro, so in our using our InfoGest model. So just to give you some practical example, this is one of the outcome of this in silico screening we have done, combining literature data together with also metabolic pathway analysis, genomic and proteomic fishing. We have analyzed several different types of plant proteins, single cell protein, and insects. And we have a different level of information, like for lectins, we can see that they may possibly be quite ubiquitous molecule. They have metabolic reasons for being actually expressed both in plants but also in single cell proteins, and the presence of lectins could be collected mostly at genomic level, meaning that we can say that potentially there could be this protein expressed. We have some type of anti-nutritional factors that are food dependent, meaning that, for example, chitin as a non-digestible carbohydrate may be impairing the digestion of protein in insects. To a lower extent, in a slightly different form, we can find them also in fungi, but predominant, this would be a non-digestible, this would be an anti-nutritional factor in insects. And when we look at the lower molecular weight anti-nutritional factors, we can see, for example, phytates, phytic acid, to be quite ubiquitous in different ingredients. So this helped us basically move forward when we had to start our in vitro analysis. And this is where we started facing challenges. And one of the first things we need to understand is that we do eat ingredients. Well, actually, we do eat foods containing ingredients. But when we want to analyze this anti-nutritional factor, we work with extracts. And this is rather important because when we approach the screening, this proteomic and metabolomic screening we have done to collect as many information as possible using mass spectrometry, we could use quite harsh extracting buffet. We do not need to keep the native structure of the proteins or to keep the molecule intact. We just want to get as much as we can. And these harsh extracting buffet are very useful in this respect. And so they can provide us quite of a good understanding of the ingredients. Nevertheless, when we want to measure the activity, so the inhibition, for example, that molecule can have in a physiological environment, like a gastrointestinal digestive system, well, we cannot use this harsh extractant, but we heavily rely on preserving the native conformational state of the molecules. So we need to use physiological extractants. And this is where we have to balance our decision, as actually extractants, the method need to be aligned to the matrix complexity. For example, we need to have different approaches, whether we are working with pulses or whether we are working with single-cell proteins, as microbials and fungal sources, they actually have cell walls. So this is where physiological extraction needs to be associated with bit milling, for example, or may require homogenization phases at the beginning. So I'll take you through a few examples, starting with phytic acid as an example of a small molecule. So what you see on the left-hand side of the screen, this is the structure of the phytic acid, where we can clearly see that there are 6 phosphate groups that are negatively charged, and these work like they are magnets, and they actually bind to essential cations, like can be iron, can be calcium, can be magnesium, and they actually, once they bind to these divalent cations, they block them. They work like they were a cage, and they actually create insoluble complexes, and they reduce their bioaccessibility. Basically, they are not bioavailable anymore, and these cations cannot be absorbed. So the presence of a high level of phytic acid in the diet can actually, in the long term, cause mineral deficiencies. So when we started looking at the different methods that are available for measuring phytic acid in foods, we found several methods, and actually the pioneer method, one that is very widely used, as is very simple, is the weighed reagent-based method. Nevertheless, this is a method that causes quite high variability in the results. There are more recent developed methods. We have, for example, the ion exchange chromatography-based quantitative method. In this case, this is also a standard method, is a validated method that would monitor the more active form of phytic acid, and there is a more recent method that should be more specific, as it is actually using an enzyme, a specific enzyme, looking at the phytic acid in its most active form, meaning that it has the 6 phosphate groups, specifically overcoming some of the limitation of the ion exchange chromatography, where there is coelution of different forms of phytic acid, more active with low to no activity forms of phytic acid. So one issue when we have different methods, and this is actually true for everything, is even true for quantifying proteins in a given ingredient, is very important that we always trace the amount of a molecule we are quantifying to the method that has been used, because the method is what would make that quantity comparable across different type of ingredients. And so it's always important to not only provide the quantity, but also provide the method that was used for that given quantity. And of course, a step forward would be the harmonization of all these different type of methods, trying to provide a common quantity. So we also have looked into, as part of the Giant Leaps project, methods to reduce the level of phytic acid in the ingredients, even in processed foods, through fermentation. There are thermal processes that can actually lead to the reduction of phytic acid in this active form. Nevertheless, when we start breaking down the phytic acid to forms that have low to no activity, these methods, they need to be supported by a certain level of validation in vitro, where the bioaccessibility of divalent ion of the processed ingredients of the processed food should always be validated in comparative studies where untreated control are used. So we actually do not measure how much left active phytic acid we have in the ingredient, but actually we try to see how much of the divalent ions are still bioaccessible at the end of the simulated gastrointestinal digestion as compared to the untreated sample. And this is just very rapidly an example of phytic acid screening in different pulses and cereals ingredients. These are not only flowers, so we do also have protein isolate and protein concentrates from pulses, which are becoming quite mainstream ingredients in the production of these alternative foods. And what is quite evident is that phytic acid is enriched in isolated proteins ingredients, and this is due to the processing. This ingredient goes through for the fractionation, as we know that proteins are solubilized in an alkaline environment and then are precipitated through acidic precipitation, and actually phytic acid solubility mirrors that of proteins, and so is enriched in the alkaline environment and crash out with proteins through acidic precipitation. Let's move to enzyme inhibitors, so proteinaceous anti-nutritional factors. So how do they work? We have here on the left-hand side trypsin and chymotrypsin. These are the most abundant proteases in the pancreatic fluids, in the pancreatic juice. And in the presence of inhibitors, which are themselves proteins, and this is a serine inhibitor, what they do basically is to block the enzyme in their active site. So once this enzyme is blocked in the active site, it is not capable anymore of catalyzing the hydrolysis. So they impair protein digestion. We cannot digest any more proteins, and this is true also for amylase inhibition. So how do we analyze and how do we test whether there is active enzyme inhibitors in our ingredient? We technically incubate our trypsin with the ingredient's protein extract, and we test the enzymatic activity of the trypsin. And what is important again is to have a protein extract prepared under physiological conditions, because we really need to preserve the native structure of the inhibitors in order it to work in our system. Another very important thing is that, as I said at the beginning, is not only protease inhibitors that can impair digestion. We also have small molecules that could impair digestion. So in order to link univocally the inhibition to the protease inhibitor, so to our protein inhibitor, we have taken a step where we have been thermal treating our protein extract in order to denature the inhibitors, so that if the inhibition was still active, was still present in our in vitro assay, this would suggest us that there are heat stable molecules that are causing that inhibition other than the inhibitor themselves. And last but not least, to ensure biological relevance, the inhibitory activity should also be tested under simulated physiological conditions. As to test the enzyme inhibition, we work with the protein extracts, while actually under simulated physiological condition, we do not remove the ingredient matrix effect. We take into account biochemical dynamics of the gastrointestinal environment, meaning that we account for pH fluctuation, but also the dynamism of the enzymatic digestion that could have an effect. And what you see here is just an example again of trypsin inhibition tested by protein extracts, and we see that there are some of these ingredients that have inhibition of trypsin. Particularly, these are protein concentrates, and this is because we have an enrichment of these protein inhibitors that are not modified by any means, because the protein concentrate is obtained through our classification, so basically is a physical enrichment. And this is instead the testing of the pancreatic trypsin inhibition under physiological digestive conditions, where you can see these are the same ingredients. You can see that the inhibition becomes negligible in the duodenum, and even after autoclaving, so means even after the thermal treatment. And the autoclaving is the most effective thermal treatment in denaturing inhibitors, protease inhibitors. So last but not least, lectins, we will only touch base on this. So we said that lectins can potentially be present in several ingredients in many of the organisms, and potentially because we have been addressing this from a genomic perspective, we have also collected later on also some protein level evidence of expression. So lectins are very complex. These are proteins that can bind carbohydrates. Why are they important? Because we have carbohydrates on the enterocytes, and so these proteins can actually bind to intestinal brush border membrane enzymes and can compromise nutrients absorptions. Lectins can be really different families, and they can be dramatically different because of the specific carbohydrates moieties they would recognize. So how do we test whether there is active lectins in our matrix? Again, we need to start off with physiological extracts of proteins. So we need to solubilize our proteins in physiological buffers. And we use something that is called erythroagglutination assays, where we incubate our lectins with red blood cells. And whether the lectins are present and they are active, they would start cross-linking specific carbohydrate moieties on red blood cells. And basically what happens is that the negative control, we don't have lectins, we would see actually a spot, a red spot, which you see here at the bottom of our well, while actually if the lectins are active and they start cross-linking carbohydrates, we would start seeing actually a network, and this is what you would see here, which is a cover that on the well that would suggest the presence of the lectins, and this method is also semi-quantitative. So just to summarize, for advanced identification, the coordination of all this information is basic. Integrating phylogenetic analysis and omics would help moving beyond the basic analysis, which would make the information way more informative and useful. And also the development and validation of multi-analytical approach for active anti-nutritional factors quantification is actually crucial. Because of this issue and the protein solubility and bioaccessibility, we really need the strong methods to be able to test these anti-nutritional factors in the alternative ingredients, like, for example, single-cell proteins, or even more new ingredients may come up from the from biotechnology. And we have seen that actually even processes like fractionation or processing can actually influence anti-nutritional factor profiles. And so the use of in vitro studies for simulating gastrointestinal digestion and understanding the effect of fractionation and processing on this anti-nutritional factor would help validating protocol for effective anti-nutritional reduction. So thank you so much for the attention.
Excellent. Thank you, Chiara. So I hand over to Corentin. Chiara and Claire, there are a number of questions in the box. You can actually start typing some of the answers because most of them are about the risk assessment, and we can go through them after last talk, after Corentin's talk. Okay? Corentin, that looks good. Very good.
Okay, thank you, André. Good afternoon, everyone. I will try to add the last layer of this session. And in my presentation, I would like to take you through how do we measure actually protein bioavailability in vivo in humans by the means of stable isotope. And for this task, I will use the example of fababine, which is a paper that has been published 2 years ago, if you want to have a deeper look. So I'm actually a postdoctoral researcher working at INRAE AgroParisTech in the research unit of nutrition physiology and eating behavior. And I will go through the presentation. So before we go to the method of measuring protein bioavailability, I would like just to refresh you a bit with the concept of protein bioavailability. First you have the protein intake. So here we take the example of faba beans. So the protein from faba beans, as was said today, earlier today, they will enter the stomach. In the stomach, you have the combination of acid and pepsin that will contribute to protein hydrolysis. And from that, you will have the formation of peptide and polypeptide from the initial protein, and some free amino acid will be released. Then the digestion will continue in the small intestine. In the small intestine, you have the secretion of pancreatic enzymes, and you also have brush border enzymes that will continue to chop the peptides into amino acid, but also, as Linda said, in some di and tripeptide. And these smaller molecules, such as amino acid, they will be absorbed and will enter the bloodstream. However, some of the amino acid will Some of the amino acid will escape digestion by the end of the small intestine and will end up in the colon. In the colon, you have a microbiota that is capable of fermenting this protein breakdown product, but the product generated by the microbiota will not be directly used by the host for its own needs. So we consider this fraction of protein to be lost, and we call it the digestive losses. Of the amino acids that are absorbed, most of them will be used for the body requirements, such as protein synthesis. But of this absorbed amino acid, some will be catabolized and transformed into urea, and will be eventually excreted in the urine. So these amino acids that are catabolized are lost, and we call it the metabolic losses. So this leads me to the definition of protein bioavailability as we quantify it in our in vivo studies. This is actually the protein intake minus the digestive losses, so the fraction that is not digested, minus also the fraction that is catabolized and transformed into urea after absorption. So in the title of my presentation, I also mentioned that to quantify protein bioavailability, in our case, we use a stable isotope. So what are stable isotopes? They are non-radioactive atoms. And here is the example of hydrogen. So in nature, the most common isotope of nitrogen is protium. That's the hydrogen we find mostly, and its atomic weight is 1 because it's composed of 1 proton and 0 neutron. And then there is the heavy isotope of hydrogen, which is called deuterium. And this deuterium has an atomic weight of 2 because it contains 1 proton and 1 neutron. And in nutritional studies, but also in other biological studies, those stable isotopes, such as 15N, 2H, or 13C, are used to track molecule of interest in the biological system. And in our case, it's in the human body. Today, we are focusing on protein bioavailability. So the molecule we are actually trying to track is amino acid. And you can label this amino acid with stable isotope at different positions. You can label the carbon. You can also label the hydrogen or the nitrogen. And in the case of Giant Leaps, we labeled the nitrogen atom with 15N. So how do we label actually the amino acid and the protein of the plant of interest? So in this case, faba bean. So here is a schematic representation of faba bean that is growing. And you also see the picture that was taken at Teagasc. So what they did is that they sprayed the faba bean with a fertilizer that contained the stable isotope 15N. And by spraying this isotope, the plant will integrate this isotope into its protein. Sorry. And how does that look like? Well, here what you see is the amount of atoms that are actually 15N atoms out of all the nitrogen atoms for the label fababine. So here we reach a 15N atom percent of 1.2. And you may wonder whether that's a lot or not. Well, if you compare it to the natural abundance, so how much 15N atoms would you find in nature without enriching the system, you see that it's around 0.4. So it means that we actually multiply the enrichment by three times. And this is sufficient for us to track the protein of interest during digestion and after absorption. So once we have labeled the food that we want to test in our in vivo study, we have to place a nasoileal tube in our subject. So the nasoileal tube is a tube that goes from the nose until the end of the small intestine. And as I said in the first slide, we target the end of the small intestine because that's where we consider the enzymatic digestion by the host to be completed. And if you look at it with an X-ray, this is how it looks like. So you see here the tube in a subject, and it starts making loops because it's actually following the shape of the small intestine. Then if you zoom at the end of the tube, this is how it looks like. So we have at the end of the tube a ballast. The ballast is actually a way to give some weight to the tube, and this will help the migration of the tube throughout the gastrointestinal tract until we reach the target position. Before this ballast, we have a collection zone. So the collection zone is simply a tube with some holes. And with those holes, we are going to pump out the digesta from the intestine. Before this collection zone, so a few centimeters before, what we do is that with another tube, we are going to infuse an indigestible marker, in our case, polyethylene glycol, and we are going to catch back this indigestible marker with our collection zone, and by measuring the dilution of our polyethylene glycol in the digesta, we will be able to quantify the intestinal flow at the collection zone. So we have an idea of how much digesta is passing at the collection zone, and we can determine the amount of nitrogen that is escaping digestion. So this is our sample collection scheme. So the day before we start the experiment, the volunteer, they come to the hospital and they are intubated by a trained gastroenterologist. We check the position of the tube by radiography. At around 8 p.m. The volunteers, they receive their last meal, and then they go for an overnight fasting to empty the proximal gastrointestinal tract. And in the morning, around 10 a.m., we feed them the test meal that is labeled and contain the protein that we want to assess. And then for 8 hours, we are going to sample different types of samples. We are going to collect the digesta. And with this digesta, we are going to quantify the digestive losses. With these digestive losses, we can determine the digestibility of nitrogen. And nitrogen here is used as a proxy for protein digestibility. And we also measure the digestibility of single amino acids. With the urine and blood sample, we are going to look at the metabolic losses. And those metabolic losses, as I said, they are representing the fraction of absorbed nitrogen that is catabolized and transformed into urea. And with that, with the digestive losses and the metabolic losses, we can calculate the bioavailability of a protein of interest. So this is a video of a real live intestinal sampling. So you see the volunteer that is intubated with this nasoileal tube, and we are just pumping out the digesta from the body using a syringe. So once we collected our sample, so as I said, we collect intestinal content, blood, and urine. We are going to analyze them in the lab. And in our case, most of our analysis, they rely on isotope ratio mass spectrometry. And this kind of equipment enable to determine the ratio between 15N and 14N atoms in our samples. And from that ratio, we are capable of telling how much dietary and endogenous protein is in the sample. If we have a closer look at how the method works, so here you take a sample, in this case, intestinal content. It can also be blood or urine. And these samples contain a mixture of dietary protein and endogenous protein. So the sample will first go through an oven where a combustion will occur, and this will lead to the formation of nitrogen oxides, and this NOx will be reduced and will form N2. So this N2, we are forming a gas. This gas will be transferred towards the ionization source. And in this ionization source, we are going to impact the N2 with electrons in order to form ions. And during this ion formation, we will obtain 14 and 15 N2 ions. And these ions will be accelerated, and then they will be directed towards the detectors. And on the trajectory, we have a magnet that helps to separate the two ions, and it will also focus the ions to the associated detector. And by measuring the quantity in each detector, we are able again to obtain our ratio between 15 and 14N. And with that ratio, as I said earlier, we are able to quantify the quantity of dietary protein in our sample. So with that said, we are going to look at the digestive losses. Here, what you see is the digestive losses for dehulled and cooked fababine. So the graph that you see here is the dietary nitrogen flow rate at the end of the tube, so at the collection zone. And so when we start feeding the volunteer, so at T0, there is no dietary nitrogen because the food is just ingested. And slowly you will see that the dietary nitrogen will appear at the tube. And at some point, there will be a decrease because digestion is completed. So this dietary nitrogen flow rate is representing the amount of dietary nitrogen that is escaping digestion. So if you sum all these points on the curve, you can calculate what we call the digestive losses. And in the case of dehulled and cooked fababine, they represent 16% of the ingested nitrogen. And if you subtract these digestive losses from the ingested amount of nitrogen, you can calculate the true ileal nitrogen digestibility, which we also call the true protein digestibility. And for fababine, 100 minus 16 give you a digestibility of 84%. So I just mentioned that we do this quantification of digestive losses for the nitrogen to get to the digestibility. And you can apply the same principle for every amino acid. So you can measure the digestive losses of each amino acid. And in this way, you can calculate the true ileal amino acid digestibility. And with this digestibility, you can calculate the score that André mentioned in his introduction, the Digestion, which stands for the digestible indispensable amino acid score, which is the score recommended by the FAO to assess the nutritional quality of protein. So now we've looked at the digestive losses. Now we are going to look at the metabolic losses, which represent the absorbed nitrogen lost through urea. And in this case, we are looking at two pools, the blood, which is actually a proxy for the total body water, and the urine. So I'll come back to that in the next slide. So here, what you see is the amount of dietary nitrogen recovered in body urea over time. And you see that at start of the digestion, there is no nitrogen from the food catabolized into urea in the body. And this will increase throughout time, and then it starts stagnating or even decreasing. And as I said here, we measure this body urea in the blood, and we extrapolate to the rest of the body to quantify the total amount of urea from the food that is present in the body. And you see that for fababine, after 8 hours, we still have 12% of the ingested nitrogen that is present in the body in the form of urea. And we also have another pool, which is the dietary nitrogen in urinary urea. So actually, this pool here from the body is being emptied in the urines. And you see here the cumulative recovery of dietary nitrogen in urinary urea for the 8 hours after feeding. And you see that at 8 hours, we recovered 6% of the ingested nitrogen in the urine in the form of urea. So if you want to quantify the metabolic losses, what you have to do is quantify the amount of dietary nitrogen in the body urea after 8 hours that is still present. And you also add to that the cumulative amount of nitrogen that you actually recovered in urinary urea over the 8 hours. So for our fababine example, if you add 12 and 6, you get to 18%. So the metabolic losses for fababine here represent 18% of the ingested nitrogen. So this leads me to the conclusion or the calculation of the protein bioavailability using 15N stable isotope. What we saw is that we measure the digestive losses, and for fababine, it was 16%. And we also quantified the metabolic losses in the body and in the urine. And for fababine, it represented 18%. So if you plug those numbers in the protein bioavailability formula that I showed in my second slide, you can calculate this number. And if you subtract 100, so the protein intake minus the digestive losses minus the metabolic losses, you get to a protein bioavailability of 66% for the fababine. So with that said, I thank you for your attention.
All right, so I would like to thank all speakers. We have a bit of time. We have about 10 minutes, and we have a number of questions. If you wouldn't mind showing yourself there, we might start with the easier one. Linda, is that okay? You typed a response in there. I just read out the question. As bioavailability is a major issue for food peptides to harness their full biological potential at the target site inside the body, what real world strategies can be employed to enhance their bioavailability and accessibility and stability. And Linda already typed an answer there. Is there anything else you want to add, Linda or Corentin, for that matter?
I suppose all I wanted to say is that we are looking at how protein in food is digested, how it's absorbed, and how it's bioavailable. We're not looking to extend its half-life in the circulatory system, to extend and make sure that it is bioavailable and reach the target organs would be something that the pharmaceutical industry has been working a long time at, and they would have things like Ozempic, which is a GLP-1 peptide, and their mechanisms for extending the half-life, the circulatory half-life of peptides includes amino acid modifications, adding fatty acid chains so it binds to albumin, maybe covering it in PEG. These are strategies that are used by the pharma industry, but we're looking at how proteins in food are digested.
Yeah, that's okay. Thank you, Linda. If you have any further questions, even after the webinar, either send us an email or you can also type it in the YouTube channel if you wish. I just go to the next one, Chiara. It is about the risk assessment of anti-nutritional factors in vitro. So you typed your answer, an extensive answer. Do you want to add anything to that, Chiara?
Yeah, so just, yes, I can add that actually, when we approached this we approached this issue, we started looking at mainstream ingredients that were already used, like soybean as being a comparator. But what we realized later on is that when we look into anti-nutritional factors, we actually needed to look at those foods that of that anti-nutritional factor would have high level, and use those ones as benchmark. And this is what I just added, as would be lectins in beans, in common beans, or protease inhibitors in potatoes, which are quite rich of that.
Yeah, very good. There's another question for you and Claire from Jessica Schwartz. For evaluating allergenicity risks, what do you think about using size exclusion chromatography for characterizing digested peptide size? Claire?
Yes, I wrote a response to Jessica in the chat, but basically I think GPC is a useful method because you can retain disulfide bonds, and if you do it under physiologically relevant conditions, if you've got non-covalent interactions that hold peptides together in larger aggregates, that is very useful, and you might want to couple it with other types of analysis, whether that's SDS-PAGE or mass spectrometry, but I think it is a useful method. And it's been used to characterize hydrolysates in allergenicity before.
Yeah, very good. There's another question by Sophie Kendler. How does EFSA include the in vitro digestibility and risk assessment in their recommendation? Are those recognized methods? So I basically typed in the response in terms of the digestibility, so the in vitro diets and digestibility that is happening at the moment. So the paper is published, the ISO method has been submitted, has already gone through 2 rounds of revisions. So I'd say it will be published within this calendar year. And Claire, you mentioned that food allergenicity risk assessment, that there are several guidance documents, and you can also look at opinions. Is that correct?
Yeah. So if you look there, I put the guidance links in the chat thing.
Oh yeah, you did. Oh yeah, I see that now. Yeah.
Because there are several there, and I think you need to go and ask EFSA.
Yeah. Very good.
But there is a lot out there, and it's what you always have to remember is it's a scientific process, and you need good science. And if it's something that you can get, you need scientific, high quality scientific approaches. And if you stick with that, you'll be all right. It's when people try to cut corners and the science isn't sound that there are challenges and difficulties.
Yeah, very good. So just to mention, at the peak of the webinar, we were about 80 people present live. So that's very good. Thanks for everyone, sticking to the very end now. So we have 3 more minutes. There's one question by Antonio Fernandez Dumont, interpretation of outcome of GI models in the context of risk assessment, so protein toxicity and allergenicity prediction might be challenging. Oh, yes, they are. Expert judgment is crucial, but what do you think about developing criteria for the interpretation of GI models for risk assessment purposes? Would it be possible? Thank you for this event, very informative. Claire, you started typing there already. Do you want to add anything?
Well, it really, I mean, it is a technically challenging thing for the toxins, and it depends where you're coming from, because the way in which the digestibility is used in, for example, newly expressed proteins in genetically modified organisms is slightly different, because there you have things like Bt toxin that are actually introduced into the plant. And if it's there in the edible tissue, understanding how that gets broken down. And I actually think that there are ways you can actually use the methodology that we've got and to calculate half-lives and look at persistence and put numbers on there to actually say, well, how much gets exposed to the mucosa. You can actually do those calculations. And I think that would be a very interesting experiment to do, a thought experiment based on information that might be there. Some of it is not in the public domain, though. For some of those, they would be proprietary to the ag biotech industry. So I think you can do that for that. That's quite simple. I think for allergenicity risk assessment, and actually Antonio has written specifically allergenicity prediction, I think that this is much more complicated, and we already know that allergenicity is a feature not simply of the material that you're giving to someone, but a feature of that person's physiological predisposition. And I think that is much more complex to actually interpret. But it might be that if you do have foods that are very, very readily, or molecules that are very readily digested, they will pose a lower risk in the same way that, otherwise you would not. I mean, hypoallergenic, extensively hydrolyzed formula, this is used for kids who are allergic, and you can make a hydrolysate that still has significant peptides left that actually they will not react to. So, I think there is a way there through of at least saying this is very low risk. This is the one you go to the next step. I don't know whether that helps, makes any sense to you, André.
Just about, Claire.
It is complicated.
Yeah. I just have one last question for Chiara. 1 minute. Yeah, 1 minute. That's a question from Andrea Rivera del Rio. Have you also looked into the role of polyphenols as anti-nutritional factors?
So yes, we have been quantifying and profiling polyphenols in these ingredients. Nevertheless, the ingredients that were analyzed within the project did not contain very high level of polyphenols. So mostly these are fractionated. So within the context of Giant Leaps, I cannot say that we have done that in separated projects, which are side projects where we have working primarily with leaves. For example, we have seen quite dramatic reduction in digestion.
Very good. Okay, I think we are done. I would like to thank all the presenters. Really nice talks, and everyone still present. There are about 60 people present now between Zoom and YouTube. Thanks for your patience and sticking around, and thank you all for your attention. Okay, bye-bye.
Bye.
Thank you.
Thank you.
Bye.
Bye-bye.
Bye.